Hi, and welcome to the fifth and final class for R for lab work. So today we're going to actually be looking at bioconductors or workflows. So bioconductor not only has packages to run data, you also have a very well described and well developed workflows to work with certain types of data sets. So let's go over here. So I have the link for this right here. All right. And you can see there's workflows here. There's only 30 of them, but they're rather useful if you use them and using with the right data flow. So if you want to look at the workflows, they have a whole tab right here, and you can look just like the packages. You can search them and 0. You can search them, or you can look at the ranks, and then look at the various descriptions. Today we're going to be looking at gene MX workflows as an example. So you click on this, and it'll take you to this page essentially. And it talks about, just like the packages, it has a bunch of information about this and how to use it, how to install it, documentation for it, but also there's documentation here of we'll get to that in a second, like the script and the text and the license and a bunch of other information. And this is the basic script. You can see it's just a lot of details, but then code form in R. This is not what we're going to actually focus on today. Now that's the same thing. We're actually going to be looking at the HTML, which once again is in these links I have inside the example code. And today I'm actually going to be very much just going through this one at a time. And you can see it's very extensive data processing tool. It's so much that I'm not going to cover all of it today. I believe I only get to about here. This is when I stopped working on it because it was a little bit too much. Yeah, it's a data workflow to analyze. So I'll just read this. Here we walk through the end-to-end GeoMX next generation sequence gene expression analysis workflow. We start with the raw gene expression. Count file is used in a combination of nanostring development and open source R packages. We evaluate the samples and express target and parent gene level count data for downstream analysis to understand our spatial data. We perform unsupervised clustering, dimension reduction, different genes, and expression analysis of visual and explore the results. And it's basically a paper. Here's the title, and here's all the people who worked on it. I recommend going to look at this yourself. Without all that out of the way, let's actually get into the code. This is the title and the people who run it. And the motivation is to basically show workflows how they work with GMX and next generation gene expression data. So it is very specific, but it is very effective. And it was just me showing you an example of how to use these workflows. So just basically just read through it, and it's very one by one. And it's basically run all the code they show us here while we're going through it and explain a little bit of it. So I took from this and put it in our shell so we can run it and look at what it does. But if you want to know more about this one in particular, you can read up on it. This is just an example. I don't know which type of package you would actually want to use, so I'm just showing you a good example. All right. So let's install and load the GM packages we need. And we do that by installing the bio C manager if not present. So let's do that. And it seems like I already have it, so that's good. Install. And for this, you can say all. Let's get everything downloaded that I need. Okay, and since I've recently, let's go in this download really quick. All right, and then you see since it's said stop, no longer has it. It's fine. It's complete. Okay. And you can see, well, actually, went way too quickly. And just keep on installing everything you need. Sorry. Oh. Okay. Sorry, it just got into a bad loop. Well, everything. Keep on downloading everything you need. We're pressing all, which is A. Okay. And then finally, we're going to run the libraries. Basically, we installed everything we need into our R program. Now we're actually going to bring into the specific like instance of R by using the library function. Cool. So to check if the current version of the workflow are being used, if the warning message appears, download, update, or the package, then reinstall it again with the above code. So basically, we're just making sure that everything is. This is basically testing if like you have everything updated, so you actually can use this workflow. And since no warning message came up, it means everything is updated. If a warning message comes up, you have just follow the instructions that are right here. Do the same thing again. No warning message, so we're all good for these versions that it's checking. So we're going to load the data we're going to use. In this vignette, we will analyze genomics kidney dataset created with the human whole transcriptome atlas WGA assay. And then here's the download link right here. So we're going to actually just download the file directly into R using the file.path. And I already downloaded it onto my instance. It's like right here. So we're going to download it onto RR as data directory. Sorry, by file path, this is actually the file path directory. And then right here, we're actually going to stitch the file path together and get all the files from it. So now we have the files. This really quick. So we have this file path, which leads into here. This file path right here leads into this part of the folder. And then this directory file path code. Essentially, it takes the file path right here, this one that we just assigned, then adds this to the end, so we get this folder right here, the CCS, and then it starts looking, and in that folder, it starts looking for this type of data, the DCC, and then look for full names, true, and recursive true. So basically, it's going to this folder, it's finding this subfolder, then it's looking for every single file in this folder that's .dcc, and then putting it inside this DCC file directory. And it's doing the same exact thing for the PKCSs right here. So it's just making directories for the various files that we actually want to take off our computer later to analyze. And then this right here, Annotation is doing the same thing, but for this Excel file that I was just doing over here. So basically, it's just figuring out where the files that it's going to use are on your computer so it can access it more easily. We load data to create a data object using the read nanostring GMX set function. So this is going to take all the data that are in these 3 folders and then put it into like one big data set. And you can see it's assigning the DC files from the DC directory, PKC files from the PKC directory, the peanut data files from the sample annotate, and then like these are just settings that you can do right here. All the expressions, annotations, and probe information are now linked and stored together into a single data object. All right, so it's all together. Now all these different files are all one object now inside the R instance. First, let's access the PK files and ensure that the expected PKs have been loaded. In this study for the demo data, we're using the HSA WT PK files. So first thing we're going to look at is this knitr library, and what knitr does is that it just helps make like tables that you can see inside the console. You don't have to really worry about it. Like you'll be using it to like look at things, but you can learn it later. And then we're just going to annotate the demo data using the annotate function, RKS. And then we're going to use the gsub function, and R will substitute the string or the character in a vector or a data frame with a specific string. So it's going to replace this gsub, this string, which is PKC, with just blank, with nothing. So you're just deleting it functionally from all the data, from all the PKC data we just made. All right. And then we're going to actually make a table. And you can see that we do have the data. Very roundabout way to basically be like, okay, this data is in here. We see it. It's been uploaded into the R instance. And this is once again the function to make tables, little tables that you can see inside your module. Very useful for R Markdown and things like that, but not really useful for like, it's there to like see things. Never useful actually programming. Now that we have loaded the data, we can visually summarize the experimental design for our dataset to look at the different types of samples and ROI and AOE segments that have been profiled. We present this information in a Sankey diagram. Load libraries for visualization. So we're going to load these various libraries for visualization really quick. There you go. Assign column names for the snaky diagram. So we're just going to set up the column names for the snaky diagram. We're going to create. Count observations by the group. So, and then pdata extract phenotype data from the ball gown object. So we're going to extract the phenotype type from the phenotype type from our demo data, and then we're going to count it. All right. Assign Sankey figure columns and links based on the column names. Just going to give them column names. There we go. And then we're going to bind links together with rbind. So links equals rbind, and we're going to join these 3 columns together. Models create nodes at the identifier of the unique values. So we're going to basically extract only unique values that the unique phenotypes present in the nodes. So we later can sort things into those nodes. And then we're going to take the syncing network is base 0, not 1 based. So that's unusual for R. So to correct for this, you need the -1 from it. And then now we're going to create the Sankey figure, which is what actually is more interesting. So here's the Sankey figure. So you can see it's interactive, which is interesting and cool. And you can see how they flow into each other. Disease 1, 2, 3, 2, 4, and then see how they actually interact. So that's cool. Cool little representation of the phenotype and the genetic, and like the result. After that, we're going to move on to something different. Before we begin, we will shift our expression count with the value of 0 to 1 to enable downstream transformations. Shift counts 0 to 1. All right. And then default QC cutoffs are commented in brackets adjacent to the represented parameters. Study-specific values were selected after visualization. The QC results in the more detail below. So this is a cutoff of like, so we're looking at various sequencing data, and these are like the cutoffs are like for what you expect from the data. So if it's below one of these figures or above one of these figures based on one of these settings, you can essentially eliminate that piece of data. So it's data cleaning of the sequence data. All right. And then we're going to take this parameter for data cleaning and actually apply it to the demo right here with this set segment QC flags. So it's going to flag any sequence that doesn't actually meet this cutoff QC, the parameters for this RQC quality control with this assignment right here. So let's do that. And then we're going to collate QC results. So we're going to do QC results, but with only the results that actually pass QC, the quality control. And then we're going to do the ones that assign that a column name for the QC results. And then we're going to summarize the QC results by creating a data frame. And we're going to actually like use if else to like adjust the data frame we've created. And continue processing of the data frame. We're going to get ggplot to start visualization, assign by column. And then we're going to create a QC histogram, like the QC results histogram. All right. Now we explore each QC metric for the segment trim table. And there you go. I'll zoom in on that. Show you. Here are the QC results. The results have been through QC. And you can and these are just various types of the various QC like parameters that being shown in graphical form. So alignment and stitch and trimmed and saturated and area and nuclei. So it gives you interesting visualizing of the QC results. Now we're going to calculate the negative geometric means for each molecule. This is the variable we're making easy by, with the negative control subset with our data. with the groups as module and then the function as function, and a bunch of other parameters. And then we're going to assign the negative geomean and explicitly copy the negative geomean from the data as data to the p data. Going to do that and get this new table of the neo-geomean data right here. And get a table of the NTC count, the segment number. And then the table for the QC summary table right here. There is very much more to this module, but I didn't want to cover it too much because it's very long and you might not use it. I just want to show you an example of what type of things that Bioconductor can provide, especially in the workflows. Like it shows like a very structured way to produce data, and you can be like, oh, this seems like a lot of work, but if you just make all this, for example, all this stuff right here, I'm just gonna clear all the data, yes, clear, yeah, clear all this, clear all that, and I'm actually just gonna run all of it. Oh, sorry. I'm going to select all, then run. It's going to run everything. Sorry, probably shouldn't run that part. I'm going to restart this. I should just avoid this part right here because it's just asking me over and over again if I have something, which I shouldn't do. So I'm just going to actually do the code that does not include the installation because it will just ask over and over again. All right, and then run. Interesting. It was working earlier. I don't know why it isn't working now. Just stop this, reset this. So file path, yeah, we have that. Yeah, we should have everything we need for this. Hmm. Over here is like the how much data your R is using. Now we're waiting for this program to run. You can see it's using this amount of data currently, and then over here, it's really hard to you don't see it often, but there was a stop sign here. So if you're running programs and you want to stop it when it's like mid run, you can always hit that stop button. It's like an emergency stop. All right. Sorry, this took a moment because it was working with a lot of data. But you can see here, I didn't have to click through everything individually. But suddenly we have all these beautiful figures. I'm showing this one. And these various tables right here. So you can just run this and then get everything, all the pieces of data you actually need off of it. Cool. If you're actually interested inside this module, you can always just go read this whole article. It's like a really well written, and they have more examples here. Article, and it goes way into more depth because I think you stopped like right about here. That's where I stopped actually making programs. So it's only halfway through. This module, this workflow has a lot more to offer. That being said, let's go look at some other workflows. So the most popular workflow look at Builder facility developed a conjugate with UCSC browser track infrastructure area available for transforming data in our range of formats, so you can just read what these things do, and you can kind of tell by like just looking at them. We walk through a end-to-end gene level RNA sequence difference expression workflow using Bioconductor packages. We'll start with the fast disk files, show how these were aligned to the reference genome, and prepare the count matrix with tallies and numbers of RNA sequence. So, and it's just like before. You just click on this, and there's basically a paper explaining how to do it, and has code examples, and you can just copy and paste these codes and run them yourself. So all these are very useful if you need them. Really well written, really well documented, and a lot of these people are active in Bioconductor. So if you actually like ask them, they'll help you or have a question for them. All right. All right, that is everything I want to talk about with the workflows, just introduction to them to see how useful they are and how much they actually can provide. Thank you for listening, and thank you for taking these classes. The homework, do I have homework for this class? I don't. My homework is essentially go look at these packages. I want to look through these and then find one that you find interesting, and then run if you have and then run the data through them. So pick one of these and just run with it. Run with the example they give you. Awesome. Well, thank you for watching, and I look forward to your emails and your questions. And when I make another class. Bye.